Hi, everyone. I would like to invite everyone to go back to your seat and welcome back to join this session. My name is Yi-Hsien, a pharmaceutical scientist in the Division of Quantitative Methods and Modeling, ORS OGD FDA. Before I begin, I'd like to ask the attendees to hold up your questions for the talks, which will be addressed later during panel discussion. It's my greatest honor and pleasure to introduce the second session, NNF applications for oral dosage forms. The second session will focus on providing case studies and the discussions about situations where NNF can support product development and the regulatory submissions for oral drug products. In session 2, we have 6 speakers to give presentations. The panel discussions will be resumed after lunch break. Please go back to your seat. The session 2 is right now begins. Okay, I will start briefly. So for session 2, we have 6 speakers to give presentations. The panel discussion will be resumed after lunch break. Our first speaker is Dr. Fang, who is a senior pharmacologist in DQNM ORS OGD FDA. Followed by is another speaker from FDA, who is a biopharmaceutics reviewer in the Division of Product Quality Assessment, DPQA6, OPQA1, OPQ. The third speaker is Dr. Nikunjkumar, a senior director of PPPK consultancy from Satara. Followed by is another speaker from Simulation Plus, Dr. Viera Lukacova, who is a chief scientist in Simulation Plus. The fifth talk will be delivered by Rullo, who is the executive director CMC Regulatory Innovations from AstraZeneca. The last speaker will be Dr. Tycho Heimbach, who is a senior principal scientist and the directors from Merck. So following, as I mentioned before, following the lunch break, we will continue the panel discussion, which is moderated by 2 moderators, Dr. Heimbach and Dr. Rebecca Moody, who is a pharmaceutical scientist in Office of Pharmaceutical Quality Assessment, OPQA2, from Office of Pharmaceutical Qualities. And we will have 2 additional panelists joining our panel discussions, Dr. Tausif Ahmed and Dr. Essam Kerwash. Dr. Ahmed is a vice president and head of biopharmaceutics and bioequivalence, global clinical management from Dr. Reddy's laboratories. And Dr. Kerwash is a senior clinical pharmacology assessor from NHRA. Now, please join me to welcome our first speaker, Dr. Wu.
Thank you for the introduction. So my talk today is focusing on the utility of model master files for oral drug products. This is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. First, I will talk about the utility of PBPK absorption modeling for oral drug products. Then I will talk about potential applications of MMF for oral drug products. Then I will give you introduction on 2 potential examples of MMF for oral drug products, followed by considerations and expectations and conclusion and future directions. Regarding the utility of PBPK absorption modeling for oral drug products in generic drug submissions, it includes using PBPK to conduct the risk assessment of deviation of dissolution profiles on BE to support the biowaiver for immediate release or extended release products. And it also includes risk assessment of impact of particle size distribution on BE and risk assessment of non-comparable in vitro alcohol dose dumping studies. And that can be also used to identify biopredictive dissolution and support BE evaluation for GI locally acting products. And the utility of PBPK absorption modeling for oral drug products also include supporting the waiving of fat BE study for high-risk products and support waiver of BE study in subjects with gastric pH change. It can also be used to support BCS-based biowaiver and to evaluate the impact of XPINs on BE for the BCS class 3 drug products. And it can be used to justify BE study design, including to evaluate the sensitivity of using parent versus metabolite as BE analytes. Here, I would like to talk about potential applications of model master files. It can include use same model for oral drug product across multiple regulatory submissions. It can be used by the same applicant, for example, for the post-approval changes. And it can be the same modeling methodology and practice for oral drug product across multiple regulatory submissions used by different applicants. And in addition to that, and a verified and validated modeling platform for products following the same route of administration. For example, oral administration can also be considered as model master files. As we know, in publication, I referred here, and we published this publication, and there's a concept of MMF. The model master file is a framework under which in silico models or methodology practices related to in silico models are viewed as portable, reusable, generalizable, and shareable after they have undergone VNV, verification, validation. This is a schematic figure showing the development and application of PBPK absorption model. The inputs include API-dependent parameters, formulation-dependent parameters, physiological parameters. After oral dosing, and it can be absorbed through GI tract and go to the liver and the metabolism and go to the systemic circulation and a link to the drug exposure. So among these modeling components, the API-dependent parameters, physiological parameters, and also the liver metabolism and the clearance could be common for different modeling purposes. So here comes the case example 1. The systemic disposition model for this drug, the API, can be considered as a model master file. So this case example is for a drug X, BCS class 2 or 4 drug, and the product is an oral tablet. The original purpose of this model is to use the PBPK model support waiving of the PKBE study and the fat condition for high-risk products. And for the model development, first using the IV infusion obtained from published literature was used and fit to a 2-compartment model and extract the disposition parameters. And then the pilot fat study data was used to develop model for this tablet and predict PK profiles for test product and get the estimation of the absorption parameters. And the reference solubility API particle size, effective permeability value, and percentage fluid in small intestine were incorporated into the model. And dissolution data is also incorporated. This model is used the Z-factor model. And for model validation, simulation after IV infusion was conducted, and the predicted PK parameters were compared to the observed PK parameters and report prediction errors. And then the IVIVC was also built with the in vitro and in vivo dissolution profiles of 3 drug formulations after oral administration. And the simulation after this oral administration was conducted, and the predicted versus observed PK parameters are compared, and the prediction error are reported for model validation. And for the model application, virtual BE simulations were conducted between reference ROD and a pivotal test product and support waving fat BE study. For this submitted model, the reviewer conducted the assessment. The limitations were identified on the absorption model, including this model need further validation of using the in vitro dissolution, the PK data, and the fasting conditions. And this model also need to use relevant in vitro data and PK data obtained from different doses or batches with different release rate to validate and then increase the confidence on this model. So here comes the potential MMF. The systemic disposition PBPK model for this drug API can be established and after both internal and external validation. This is to give you an example of the potential model master files. As indicated in this graph, this model master file can be applied to support multiple regulatory submission purpose. This model master file disposition model can be used to evaluate impact of gastric pH on BE, evaluate impact of food on BE, evaluate impact of dissolution deviation on BE, and impact of permeability change caused by excipients on BE. Of course, these applications can be another applications. I didn't include all of them. Another case example is using the same modeling methodology to assess the impact of particle size distribution on bioequivalence. Some background information. This drug product is soft gel capsule, and the PK parameters Cmax and UC are found to be sensitive to changes in mean particle size of this drug product and the fasting condition. The applicant submitted a mechanistic absorption model to link PSD, particle size distribution, with in vivo PK data. The original purpose of this model is to use the modeling to support setting clinical relevant PSD specification. This strategy shows the modeling development and the validation workflow. Here, the initial model were established by using physical chemical parameters, log P, pKa, in silico predicted parameters, including solubility biosalt, solubilization ratio as inputs, and particle size as inputs, and the clearance were informed by the population PK. And then the model development was further developed by using multiple data sets from 3 different strengths and the fasted and the fat conditions, and also optimize the model. And then the model validation used the multiple data sets from 3 different strengths and the fasted and the fat conditions. These data sets were not used for model building and then to validate the model. And then sensitivity analysis on the key parameters, including solubility, biosalt solubilization ratio, and the particle size are conducted. So these PK parameters were found sensitive to biosalt solubilization ratio, solubility, and the particle size distribution under fasting condition, and sensitive to gastric transit time under fat condition after conducting the sensitivity analysis. And then the applicant optimized a biosalt solubilization ratio and the solubility for better model fitting and the fasted condition, and the gastric transit time was optimized for better model fitting under fat condition. Regarding model validation, as I mentioned earlier, and the applicant used the multiple data sets that were used or not used for model building, including using the in vitro PSD data and the in vivo PK data from 3 different strengths and the fasted and the fat condition for the validation. The prediction error were reported, and we found that were within the marginal accepted prediction error limit. So the applicant used this model, and then for conducting the simulations, they fixed the D50 value and the widening the boundaries of particle size, D10 and D90, and they found that only a minor effect were observed on the predicted PK exposures. And the reviewer also conducted the simulations, and we've also found a low risk of non-BE when we fixed the D50 and changed D90 over a wide range. For this case example, these modeling results support a satisfactory BE assessment of this ANDA as setting a clinical relevant 3-tier PSD specification. So I give this example is showing that this similar modeling methodology and practice as an MF can be used by across multiple applicants, and a new user could refer to the methodology used in this MMF. So here, this graph shows that the model master file for evaluating impact of particle size distribution on B can be used by multiple applicants. Some consideration and expectations. As we know, the value of model master file is in the understanding of the model structures and the parameters. And in addition to the two examples I gave you, a model master file can also be a master file of a set of compound and systemic parameters in the platform or a user-friendly interface. And the model master file should be classified considering the purpose of this model master file. And in general, the party who intends to share a model and make it reusable to others can be the owner of this model master file. And this owner can permit one or more applicants to incorporate this model master file in their own submissions. And of course, more than one model master file can be used for the same regulatory purpose. As we know that from the previous presentations, FDA may perform a technical review when the model master file is referenced by an application. In conclusion, as I give you the examples, the one example is the same model for oral drug product can be used as a model master file across multiple regulatory submissions. Same modeling methodology and practice for oral drug product can be used as model master file by multiple applicants. And if the model master file is implemented, used, and it will simplify the submission process, reduce cost and time for model development and validation. And of course, this model master file will support standardization in model building and V&V and improve regulatory assessment consistency and increase our confidence in the modeling and simulation approaches. Here I mentioned about the benefits. I also heard about the challenges mentioned by previous presenters. So I hope that through the panel discussion and the small group discussion, we will find some good points on the resolution and pathway forward. With that, I would acknowledge the people listed here. And DQMM, Liang, Lucy, Yi-Hsien, Sharon, Aaron, and also Liang and Rob for their valuable inputs. Reviewers from Office of Bioequivalence and the OPQ as well. Thank you. This concludes my presentation.
Thank you, Dr. Wu. Now let's welcome our second speaker, Dr. Yunming.
Thank you, Dr. Fang Wu, for the great presentation. My name is Yunming. I'm from Division of Biopharmaceutics at the FDA. I'm a biopharmaceutics reviewer. It's really nice to see a lot of familiar faces here. Today, I would like to talk about some product-specific parameters in the model. Before we start, I would like to highlight our mission at pharmaceutical quality one more time. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. This is our standard disclaimer. This presentation reflects the view of the presenter and should not be construed to represent FDA's views or policies. You probably have seen this graph so many times. When we talk about product-specific parameters in the model, we're actually thinking about what aspect of the product can be implemented into the model, and a lot of them are biopharmaceutics related. And they can be reflected by dissolution test. Right here, I highlighted some components that can be implemented into the model master file, which are the CBAs, for example, the CPP, CMAs, the particle size distribution, as well as the dissolution test. So it really comes down to two questions. One is what parameters in the PBBM are product specific? And the second question is, how can we minimize identifiability of the product while maximizing amount of information shared? Because a lot of information are considered proprietary, and we really need to be careful what to include in the MMF. Here I listed some parameters in the PBBM. And for the formulation, we have the API and the excipients. And the most important information here, I believe, it's the solubility. This is not only the drug substance solubility, but there are a lot of other aspects that can be influential to the solubility. For example, the excipients, and also the way the modeler actually model the solubility pH relationship in the model. For example, the solubilization ratio, these are usually derived or optimized from dissolution test in biorelevant medium or other complex medium, for example, medium with surfactant. In many cases, if this kind of experiment cannot be done or a biorelevant medium is not used in the dissolution test, people usually calculate the solubilization ratio or the partitioning coefficient for ionized or unionized species empirically with the equations. And in some cases, we really want people to share whether, like, how these parameters were actually included or obtained and to implement into the model. Also, for the surface solubility, this part is automatically implemented in SimSip, and sometimes people manually calculate and implement into the GastroPlus. And for this part, we really want people to share whether you consider the surface solubility or not in the model, because this will be really helpful when the applicant actually use the MMF and they want to know if this part is considered or not. And secondly, how these surface solubility were implemented. It can be implemented from the solubility pH curve, or it can be implemented into the ACAT model. You can modify the bulk pH to have the same effect. So these are all the informations we want the MMF holders to share, so it can facilitate when the applicant try to use this model for their product development. And similarly for permeability, some salts, especially the chloride salts, it might lower the permeability of the product. And some of the excipients are known for adjusting permeability for the product. For example, mannitol is known for its ability to modify the permeability. So we are not expecting the MMF holders to share what exact excipient were used in the product, but it will be very, very helpful if the holder can share, like, how these components were actually implemented, or whether or not they are implemented. For the So next, I would like to talk a little bit about distribution method, because the distribution curve will serve as the input for the distribution models, and the method will decide how the curve will be generated, and as well as the hydrodynamics will be modeled. For example, the paddle speed. Usually, the QC method will be used to generate these curves, because the QC method will be assumed to be the most discriminating. But since some of the methods are product specific, one distribution method might be discriminating against some CBAs for one product, but might not be for others. So it is very important for the model master file holders to share this piece of information, whether you use the QC method to generate the distribution curves or not, or if you develop a specific dissolution method just for the purpose of model input. And before I move on to the next question, I would like to tackle a few more things, which are the model objectives and modeling strategies. They are similar because you're not going to use the Johnson model to evaluate the dissolution curve unless you're using a PPSD approach. So for model objectives and modeling strategies, we would like the MMF holders to include the choice of dissolution model and the choice of input, because sometimes the modeling strategy itself can reflect some useful information. If you choose a multiple bin PPSD approach, it's likely to tell other modelers the traditional Z factor or refined Z factor or the one bin PPSD is not likely to work well for the model. And this will also be closely associated with the choice of modeling platform, like GastroPlus versus SimSip. So these are all very helpful information when people want to reuse the model in the future. Let's switch gear and discuss how to minimize identifiability. So the goal here is to demonstrate predicting ability of the model while not disclose exact product information. For example, the dissolution profiles, formulation, manufacturing processes, and some material attributes. These goals are not very easy to achieve because most of these are considered proprietary information. So we really need to include this information carefully. For example, one way to do this is to perform parameter sensitivity analysis or PSA to extrapolate the BE or non-BE boundary. For example, for particle size distribution, you can choose to use the Johnson model or von Flanagan model to explore the lower bound and upper bound of the particle size distribution and report that information in the model master file. And because the correlation between the particle size distribution and the PK data is likely nonlinear, so it's very difficult for people other than the MMF holder to identify the exact parameter that were used to build the model master file. But at the same time, having these BE or non-BE range, people will still have a good amount of information to set up the particle size distribution specification when they develop their products. Similarly, for the dissolution profiles, one way is to set up the dissolution safe space. Let's say if you decide to use the Z factor model and you can perform a PSA for the Z factor against the PK data and you try to extrapolate the relationship. Then include the upper bound and lower bound of the Z factor in the model master file. And because there are many parameters lumped into the Z factor or the PPSD model, it will not be necessary to disclose the particle size distribution information or the hydrodynamic model. Let's look at an example. Here I have the parameter sensitivity analysis for Z factor for a drug product. On your left is the PSA result for the Z factor against Cmax and AUC infinity. And based on this graph, you can extrapolate the upper bound and lower bound of the Z factor that are BE to the product, which is represented by the blue area in the graph. And this can be confirmed with virtual BE study. In the model master file, the holder only needs to report the upper bound and lower bound of the Z factor, like the graph on the right. And as you can see, in this way, it's very hard to pinpoint the Z factor that is responsible for the original product, because the correlation of Z factor and PK data is nonlinear. And people can still convert the Z factor boundary into dissolution curves. For example, if I would like to simulate the dissolution curve in this particular setup, it's in USP apparatus 2 in 900 milliliters of pH 5.6 phosphate buffer, we can convert the Z factor into the dissolution safe space by calculating the dissolution curve in this particular setup. The upper bound right here is represented by the red line, and lower bound is the blue line. The limitation for this method is it only works when the BE range of the Z factor is wide. In other words, if the model is very sensitive to the change of Z factor. If the model is not very sensitive, then it will be very easy for people to figure out the exact dissolution curve that were used to generate the model master file, which is not what we want. Let's look at a more complicated example. So in this case, the dissolution safe space was set up based on the virtual batches with multiple bin PPSD. The lower bound of the safe space is represented by the graph on the left with 2 particle size bins, whereas the upper bound is represented by the graph on the right with 5 particle size bins. The virtual dissolution profiles are represented by the red dots. The setup for this is USP apparatus 2, 900 milliliter of pH 5.6 phosphate buffer, and the PPSD was fitted to simulate these virtual profiles, which is represented by the blue lines. In the model master file, the holder can just report these particle size bins to represent the upper bound and lower bound of the safe space. The downside for this approach is it is naturally hard to compare PPSDs because they might have multiple bins, and it's not very intuitive to tell which one is higher, which one is lower. And if the particle size and number of bins are very close, it will be even more challenging. One way to mitigate this problem is to convert the PPSD into the equivalent particle diameter, which is the DEQ right here. This can be done by calculating the equivalent surface area of the particle size in each bin and back calculate the particle size, assuming one size of the particle is responsible for the entire surface area. In this example, the lower bound of the DEQ is 624 and the upper bound is 157. And these numbers can be included in the model master file to represent the dissolution profiles. In summary, I briefly talk about the product-specific parameters that can be used in the model. I talk about a few ways to minimize identifiability while still providing good amount of information in the model master file, which can be used for future product development. My take-home messages are share as much as you can and share nonlinear parameters or empirical parameters in your model master file. And that's all for me. today. If you have any questions, feel free to ask during the panel discussion. Thank you.
Thank you, Yunming. Now let's welcome our third speaker, Dr. Patel.
Yeah, thank you very much. As you can see, I was asked to present on practical considerations of developing and employing model master file. As you can imagine, it is probably a very tough topic for something, well, to give practical consideration for something that we are still defining, but I tried my best. So I started by reading from the previous workshop report. I think it was very helpful. This is a description of MMF I found very interesting from the previous workshop, that it can be a quantitative model or modeling platform that has undergone sufficient verification and validation. So as you can see, this one sentence, but it packs so much information in it. I think it needs a lot of discussion on each of word, which is included in this one. So I'm glad that we have this workshop today and tomorrow to go through them. I think I'm not going to dwell too much on the benefits because it has already been highlighted from the morning session. One of the things that has not been highlighted and which I believe is a potentially big benefit is that improving the consistency and efficiency through across the regulatory agencies. So I think we have been only focusing on FDA, but there have been instances where you get completely different review outcome from different regulatory agencies that probably if we can somehow streamline, I think that would be a very big advantage of model master file if it is a possibility, probably to have a harmonization. I also see that there is a potential benefit of pre-competitive collaborations, especially in establishing MMF for a context of use in a particular platform. So that can probably be another advantage that can go pre-competitive, and there can be many more. The purpose of my presentation is only going to focus on the PBPK modeling of oral drugs and drug products. So when I say model, it would be PBPK model for oral drugs. So I think in the panel discussion in the previous session, Dr. Reggie already described different components of PBPK. So I think the critical ones are the algorithms, which are in the middle, physiologically based model. So it's the set of equations in a platform, which is supported by the physiology database, which would probably be the common between different drug products, because if you are trying to treat humans, most likely many of those parameters would be common except some of the patient-specific parameters or ethnicity, as we discussed in the first session. The other big component is the drug and formulation-related parameters. So there are some drug parameters like intrinsic properties of the drug, and then some of the parameters can be influenced by the formulation. So once you all combine these two types of information with the equations, you could run a simulation. You compare your simulation, you validate or verify your model and make sure that it is predictive. Now, when we talk about MMF, how can we approach this? So again, if you look at this description in the previous workshop report, it says model or modeling platform. As a practical modeler who is doing modeling day in and day out, I think it is very difficult to separate a drug model from the platform, because the drug model is specific to platform. As Dr. Nicholas mentioned in the morning, model is not only parameters, it's a parameter plus equations. So when we say about drug model or drug product model, we have to have that model in a given platform. So probably this definition might need to be slightly modified. When we say model, it already inherently includes a platform because the equations are part of it, not only parameters. So when we go about, I think this is what I personally envision, that how we can approach MMF. I think we have to consider it as a stepwise approach. The first step, if you are buying a car, you want to look at the transmission, whether the transmission is good. So that's our modeling platform. So the algorithms and the physiological data that describes the model, are they good enough? Do they represent the most recent advances in the field? Are they reasonable for the purpose? So that should probably be the first question that should allow us to evaluate the modeling platform and its ability to simulate the scenario that you're trying to model. The second would be the context of use, which would include the model plus independent cases. So I just put one example here that I looked into literature. I found there are about 11 different drugs formulated at different products, and the SimSip-ADA model was able to predict the outcome and very different outcomes. So there are some drugs which had a strong ARA interactions. There are some drugs that are weak, some negligible. And the model was able to sufficiently predict all those 11 drugs. Now the question is this enough? Would this be considered enough to characterize this modeling platform is good enough for evaluating ARA? That's a big question, and I think we can probably discuss that in the afternoon. I don't know if we will agree to the conclusion, but that's the kind of thing. So when we say V and V, it's one word, but I think it has a lot of permutations, combination. We need to come to some kind of an agreement of what would be considered sufficient. Once we can establish that the platform works to predict the outcome of interest, which is the context of use, maybe we can go to the drug and drug product specific model. That would include specific parameters. So this is a publication from Dr. Cristoforetti et al., which summarizes the sum of the parameters that were used for ketoconazole and posaconazole to predict the effect of acid-reducing agent. So now when we talk about a drug or drug product model master file, we need to see whether the parameters are well-defined, whether the sources are sufficiently cited, the assumptions are correct. I think the assumption of the model came up quite frequently, and that can be context-specific. So the model assumption may work for one context of use, but it may not work for another one. So how can we navigate that? Because we discussed in the morning that there can be multiple modeling master files, but how are we going to evaluate which is good for which context? I think there are lots of discussions probably we need to have to make it more practically applicable. So these are some of the questions that came to my mind as a modeler. If I'm going to submit a model master file, what would be more important? So, and I think that probably, I have listed the questions. I have my own views on it, but I don't want to put it. Maybe we can discuss it in the afternoon session. So probably I would have some considerations as question. So for example, for SimSip simulator, most of these algorithms, we try to keep publishing it in scientific publications. So it's peer reviewed by the scientific community and they become available. So this describes up 3 publications of the architecture of SimSip. There is another one that how we employ the IVIVE principles or in vitro in vivo extrapolation. And the third one is oral absorption model. And then SimSip, every version, we also provide a description of, well, not every version, but for most of the data that we have, we try to describe where the data is coming from, how it influences the parameters in the software. For example, there is a documentation for healthy volunteer physiology. So we have the description algorithm in publications. There is also a database of physiology. So PBPK is not something futuristic, it is already happening. So some kind of MMF or some kind of evaluation is already there in place. And now the question is this sufficient? Would peer-reviewed publications to describe the model and equations be sufficient, or do we need something more? What should be included in MMF? Would the population summary files that I listed below is sufficient, or if we need, what should be included in those files? Where such data will be submitted. So we're talking about MMF, but who is going to submit it? How is it going to be evaluated? How is it going to be reviewed? Where it is going to be stored, updated, and distributed? So we saw that there is a particular web page for DMF. Maybe there should be something like drugs at FDA. There may be models at FDA, or maybe models at global harmonization, something where the repository can happen. Now that's modeling platforms keep evolving, so there is going to be a new version every time. So all this model documentation would need to be redone for each version. For example, some of the software can have annual release cycle. So would this be done every year? How is it going to be managed? Whether version comparison is going to be good enough, or so I think there are lots of other questions that we might have to come to discussion to make this a possibility in reality. The second one is context of use. So as I already mentioned, one literature review that there are about 11 drugs which are formulated with different drug products can be predicted nicely using the Adam model for acid reducing agent effect. Now, is it going to be enough? The other way I can look at is that some form of MMF for modeling platform for a particular context of use already exists in literature. So there is a nice publication of predicting CYP-mediated drug-drug interaction using the SimCYP simulator as a publication. There is a lot of data that is already included in this publication. And the platform providers probably can produce this type of MMFs for a particular context of use if that is a requirement, or maybe we need to discuss how it can be done as a publication, as a documentation, etc. Now, some of the questions for context of use, what would be considered sufficient? I think I already brought it up. When we say if the particular platform is good enough for a given context of use, what would be considered sufficient? Who and how would they decide what is sufficient? Would peer-reviewed publications would be enough, or do we need to provide raw data files to the regulators? Would the regulatory agencies have enough resources to evaluate all the raw files? If you have 1,000 files, are they going to evaluate it? Or the peer-reviewed publication, you are going to trust the peer review process that we have in scientific publication and use the peer-reviewed publication as a source of reference? There are lots of other questions that we can try to discuss. Where would such data be submitted? Again, that's something like a practical aspect that we need to probably figure out. Now, moving on to the third stage, and I think this is the most critical one and crucial one, the drug or drug product specific model as a model master file. Now, I think this is interesting, and I wanted to rely on what's in the public domain. So I looked into literature for ibuprofen because there are multiple publications of ibuprofen using the SimCYP Adam model. So I started looking from 2014 when the first publication came out. Then there was another one 2015, 16, and also it demonstrates which version of SimCYP they used for a particular publication. Now, in the first publication, the permeability was predicted from CACO2 data. So from in vitro, it was predicted, and the value was around 6.3 into 10 raised to minus 6. They didn't have a measure of particle size, so they assumed that, okay, the mean particle radius is 25 micrometer, and elimination was predicted from in vitro enzyme kinetic data. Now, if you look at the another publication, they used the permeability from rat data, and the value they predicted for human was almost half of the previous publication. So both of them are reasonable ways of predicting permeability, which is correct, or which is more reliable, or which is more reasonable. They assume mean particle size radius as 10 micrometer. Elimination now was calculated using the not enzyme kinetics, but in vivo plasma clearance from another source. If you look at the next publication, now the permeability was deconvoluted from the oral Ka for the solution formulation, and their value is now about 3 times higher than the other value. So now the permeability is 17 into 10 raised to minus 6, and the mean particle size was, as presented in the previous presentation, was estimated as an effective particle radius from the in vitro dissolution data. And now the clearance is from another source, which is 3.5 liters. So both the clearances are correct because this is from the public information, but one clearance, 3.5 liter from one publication is different than 1.8 liter per hour from another publication, right? Now, there are other versions of the model. I don't want to go into detail, but now this raises the question, when there are multiple values for a given parameter possible, and also they are scientifically reasonable, which is going to be considered for the MMF, or who is going to be responsible for deciding which one is an acceptable value? Even if you measure permeability using CACO2, different labs would potentially have different values. So which value is going to be reasonable? So the original developer had run CACO2 assay, the new applicant has run CACO2 assay, they are going to get a different value. So how are we going to consolidate those differences? If someone changes the MMF, what kind of validation is required to be considered acceptable? Because maybe some of the validation data was confidential to the original MMF applicant. So how are we going to manage that, the confidentiality aspect of the MMF? Again, the definition of MMF can be context dependent. So pretty much for the same drug, there can be multiple MMFs for this different context of use. So how are we going to call it master? When we call something master, that means there has to be something commonality. But if it's going to change with context, I think we'll start to lose the purpose of MMF. So that's another potential thing that, how are you going to manage that? And then the other one is that as science evolves, the MMF, the algorithms, the parameters would evolve. So if we try to maybe make it stricter, it's going to slow down the innovation in the modeling field, because we want the modelers to be innovative, to think outside the box, to make it as better as possible. But if you try to put some kind of I don't want to call it bureaucratic burden, but I don't have another word I can think of, but if you try to probably make it stringent that, okay, it has to follow this kind of criteria, it would stop people changing it, because they say, no, I have to try and do all this verification again or validation again, I don't want to change it. So it's like, if you think about this, I think I was reading the other way, the distance between the railway tracks, right? How it came up, some people say it started in Roman Empire, and because they didn't want to change it, so it's just kind of a bureaucratic transition. So it's a weird thickness. I don't know how true it is, but probably it's something, if something happens, and if it is regulated, probably it would slow down some of the innovative changes in the MMF. And again, I think this was discussed by others in the morning, that how are we going to consider different versions of MMF, especially if it is developed by different applicants and sponsors. And this is the most important question. What do we expect out of MMF? Are we expecting it to be a starting point for any new applicant to work on, or are we considered to be a final form which is not expected to be modified during the review or submission? And I think that probably would decide a lot of my other questions on the MMF. These are some of the references I use. So I just brought you a lot of questions I had in my mind. Maybe we can try and discuss it in the afternoon session and try to come to some agreement, or hopefully new path forward for the MMF. Thank you.
Thank you, Dr. Patel, for a nice presentation, and raised a lot of questions for us to consider. Now let's welcome our next speaker, Dr. Lukacova.
Okay, thank you. In this presentation, as you will see, you'll probably see a lot of what we've been already discussing this morning. When I think about MMF and what components the MMF file needs to include, I start immediately thinking about PBPK or PBBM. For one, that's my field. For other, it nicely outlines the components that we need, what we need to describe for the compound or for the specific application. We've got the drug-specific or the compound-specific properties. On the left, the system or physiology parameters on the right. The formulation or the drug product properties themselves need their own separate specific box because they may differ for the same compound and different drug products. And all of this feeds into the PBPK model, which is our collection of equations and assumptions and so on. So as already was discussed earlier, one of the principal points that I would put on the top of the list of what needs to be discussed or described in MMF is the model structure. Equations, what do we incorporate and so on. The model structure or the equations do come with the platform, right? But platforms allow flexibility. You are not going to utilize every single equation in every single model. So it is important to describe which assumptions you made, which mechanisms, which processes you incorporated in that specific model. So the assumptions then bring back to the context of use. The simpler the model, the more assumptions, the more simplifications we make, the more limited context of use for that model we will have. The drug inputs. I think this is this is pretty clear. We are looking at the drug physicochemical properties, PK description, describing the drug distribution, disposition, and so on. For the system inputs, these are the physiologies. Independent of the drug or the formulation, these are describing the physiology of the subject, and not just the physiological parameters, but also what kind of inter- and intra-subject variability we are considering for those parameters, for those physiological parameters. If applicant is using a specific platform, these are already included. Okay? But again, the platforms allow flexibility. If applicants modifies any of these parameters, it needs to be justified. It needs to be very clearly stated which default settings might have changed and why. And for the formulation inputs, I listed it here because they are part of the PBBM or PBPK model, but these most likely would not be part of would most likely not be part of MMF, but would be part of each specific application from each sponsor. This is just a brief example from the literature, and many of you in the PBBM area or who are following the PBBM applications may be familiar with one of these models for acalabrutinib. The original model was developed back in 2019 or published in 2019, was developed for acalabrutinib freebase. The model was described and validated. The little plot in the upper right corner shows the predicted versus measured AUC, very nicely validated across a number of different studies. In this case, the formulation aspects were modeled using the product-specific particle size distribution. So the way of determining this distribution and verifying that the distribution is correct has been also verified and justified for this specific product. Of course, the model described all of the different parameters and their sources and assumptions that were made for parameterizing these. In addition, they actually did modify some physiological parameters, built-in parameters in GastroPlus. I don't want to debate whether the modification was right or wrong. What is important here is that it was clearly described. So somebody else who is going to use the model would know this is the modification I need to make as well. A few years later, then they used the same model for different formulation. To reformulate acalabrutinib from a free base to maleate salt, and this was a good way of reusing parts of the previous model. What they reused was permeability and everything that happens after absorption. Assuming that's the same as for the free base, what they needed to modify was, of course, what is related to the formulation of the salt itself. And the other part that they reused was methodology. They already seen in the earlier application that the drugs or the product-specific particle size distribution was a good way of parameterizing the PBBM, was a good way of translating the in vitro dissolution into in vivo inputs, so they used the same methodology here. And again, the same modification of physiological parameter was used. Of course, the additional validation was made. I'm sure you cannot see all of those small numbers there. The plots on the left showing a bunch of error bars The first 8 or 9 of them represent the maleate salt, the new formulation showing the validation of Cmax and AUC at the bottom, and the last 2 were showing the original free base. And then the model, once it was validated, was then used to apply it to define the dissolution safe space. So when it comes to, I would now go back to model master files. What did they use in this case was more of a drug-specific model master file. They reutilized just about every component except for the formulation aspects. They reutilized everything that was related to the compound itself. But it's also important to remember what assumptions they made. In that model, the PK was parameterized using compartmental PK fitted to the adult data. Would not be very easy or even possible to extend the model to other populations or pediatrics or something like that. It's important to remember these approximations that will limit how much the model can be used. Modeling platform. It's no doubt can help to streamline even the modeling platform itself, reviewed and validated for a given purpose, can help to streamline the process. The reviewer doesn't have to go through and keep verifying the same physiological parameters over and over. But we have to remember there is more to the application than the platform. Right? It's possible to build a very bad model in an excellent platform. So the drug and drug product parameters still need to be reviewed, still need to be verified. And finally, the modeling methodology, as in the example I showed, the PPSD was used in one aspect and then verified and supported for the other application. And especially for the new methodologies that keep coming up, especially in the PBBM area all the time, I think this could help to speed up the applications so that we don't have to keep convincing 15 different reviewers from scratch that this is a good application, that it works. Once it was verified, once it was used, it could help to use for the other products. And in summary, I don't think I need to go through the summaries again. As you have seen, I only put in a bullet points for number of questions because when I started writing them out, I ended up with, similar to Nikunj, ended up with 5 extra slides with just the questions. So hopefully we'll be addressing a lot of those questions by the end of tomorrow. Thank you.
Thank you, Dr. Lukacova, for providing us great examples. Now let's welcome our next speakers, Mr. Rullo.
Thank you very much for giving me this opportunity to talk today. And I'm going to present information on a global collaborative study between industry and global regulators. In this presentation, I'll give you some background as to how the study got started, who was involved, in terms of industry members, why we even got started on it, and move on to the workshop where we shared the output of the collaborative study, give a few details on the workshop itself, give an example of one of the surveys that was conducted and how we're going to share the output of the workshop with the public. I'm not going to go through the top portion of this slide since it's been covered in other talks, but understanding the challenges that were faced, first of all, there were some authorities that had minimal experience with actually having PBBM in their applications, and while others have a lot of experience with it. Adding to this problem, industry had no common way, template for submitting their PPPMs. So you immediately had high variability in what was being submitted. You had a high variability in experience of regulators, ultimately leading to a lot of unknowns about how are we ever going to overcome some of these problems. Additionally, FDA is one of the few agencies that have a guidance in this area. And then finally, you had industry, different levels of experience and opinions as to what represented proper model validation and verification. So to address this problem, IQ Consortium got together, and a number of companies listed here agreed to participate in a collaborative study. One of the key factors that industry and regulators thought was important were that the case studies had to be real. They couldn't just be kind of fictitious studies that were assembled. In addition, there was a limitation that the case study could not be in a project where it was under development. The regulators didn't want to give any industry some advantage over some other company. So we had to take case studies for products that had been approved and on the market in all of these countries. We also had to, obviously, this map is showing all of the regulators that were involved in the studies. And since these studies were confidential studies, we couldn't share them with one another. So we had to identify mechanisms how we could submit these through use of box, as well as many other kind of avenues to the various authorities that were involved in the study. In the end, we had agreement that the industry would submit these confidential case studies. The software companies also got involved because they agreed to provide free software as well as free training to any regulator that was interested. There was also an agreement that any academic could help an authority evaluate these case studies, provided that they have a non-disclosure agreement in place. And then finally, obviously, great thanks to all the regulators that were involved who agreed to independently and then collaboratively get together to discuss all of the case studies. All of this was agreed that we were going to share this at a workshop where in August of last year, where 64 participants from FDA participated. We had 16 regulators from around the world that also participated, 22 academics, again, from all around the world, 85 industry participants, for a total of 187 in-person participants in the workshop. This is all sponsored thanks to the University of Maryland and the MCERC program. And the regulators at this meeting had panel discussions where they reviewed all of the case studies and shared what they liked about it, where they felt some of the weaknesses were. In addition, there were 13 breakout sessions where there was a deep dive into very specific topics. And there was agreement that we would publish all of the output from the workshop. I'm not going to go through this slide in detail. Suffice it to say that the day one focused on model parameterization, day two on method validation and applications, and day three on current state and new horizons. So altogether, it represented a really in-depth set of 3 days for evaluation of to try and get industry to a common place in how they went about to develop, validate, and verify models. One of the things that also happened at the workshop were a whole series of surveys. And one of those surveys was trying to see the level of interest in developing a common template for reporting. And shockingly, there was a 96% agreement from the participants that a template would be useful. Molecular Pharmaceutics has kindly agreed to published 5 publications, and thanks to the University of Maryland, who have given funding for open access, all of these 5 publications will be available to the public and also appear as a virtual special edition once all have been published. The first publication is a workshop summary, which has already been published. We're very close to submitting the day 1, hopefully quickly followed by day 2 and 3. We are in the process now of working with about 20 to 30 companies and a number of regulators as to what represents good content for a report in a submission, regulatory submission. One of the things I've learned through this process is that when I look at the number of software companies that were involved, many individuals who dedicated their time to training the regulators, the academics that were involved, inevitably, any type of acknowledgement that I put up is definitely going to miss people, because it would probably involve about 100 different people that were all participated in some capacity to make this happen. So to anyone whose name is not on there, I apologize. And thank you very much.
Thank you, Mr. Rullo, for the nice summary. Now let's welcome our next speaker, Dr. Heimbach.
Yes, how do you move the slides here? Yes, thanks, Greg, and thank you for the organizers. I know it's about lunchtime, and we had so many exciting things, so please do not worry. You will only hear about the template, and as long as you agree to the template, to look at it, you can already go. No, just stay another 5 minutes. What we have is identified as, by the way, I just have to show the slide. It means it's my own opinion, but it's still valid. So here you can see the first paper that was just published about 3 weeks ago under the guidance of Claire Mackey. This is from the workshop that happened in this same room in the late August that Greg was just talking about. So anybody who missed it, you can go read the publication, and everything is online. Again, this was hosted also by MCRC and FDA. And you have seen this. Now, why would we agree to a template? And you have to see it from different angles, in my opinion. When you work in the industry and you're filing a regulatory filing, we are in the world of global drug development and global drug approvals. So when we put a model out, it's actually that we are not just sending it to the FDA, right? It's going to other applications, other agencies, going to PMDA, it might be going to EMA, globally, any country. So that means if we submit a PBBM, we would like to have a template, because otherwise what will happen is agencies will give us a template and we have to follow it, and they're usually not the same. So that was one of the business drivers which identified the value, the business value of having a common template. The template is not intended to describe exactly how you do your model parameterization. Rather, it is to provide a framework so that when regulators or scientists look at it, they're used to seeing a certain format, and then they can better understand and more efficiently review it. And here what we have is we have basically put together a Word file that is rather comprehensive. Currently, it's about 25 pages. I will not be able to walk you through, the entire slide deck, this entire template. But as you can see, there's an executive summary under number 1. There are materials and message sections. Then there are sections that describe the model strategy. It's a little bit slow, the mouse here, but I will go through the details. So right now it's an extensive Word document. Industry can provide input, regulators can provide input, but because the framework is there, it can be readily modified. So if you break it down into a little bit smaller pieces, you will be able to see that a lot of thought has already been put into that. The executive summary highlights just that. In some cases, it will talk about the previous approval history, but also talk about the intended purpose of this particular PBBM, right? So this template is for companies filing something with the FDA, but you can imagine this particular template is also useful company internal or for other applications where you just need some structure around the context of use and what you're trying to answer. So here we have a question of interest and the context of use. In this particular case, there was just a case study from the workshop that the agency agreed that the batch not passing F2 can still be considered bioequivalent to the reference product with the QC dissolution method. And then what happens is they demonstrated that that particular QC method is not bioequivalent, it's not biopredictive. They came up with what they call a physiologically based dissolution testing and demonstrated that this particular batch is leading to bioequivalence if you run virtual bioequivalence trials with some tool like SimCip or Gas2+ or others. So the context of use, I'll come back to that a little bit later, has really not been heavily used in the side of PBBM, focusing on drug product quality and more the physical pharmacy applications and dissolution data modeling. The context of use has had traditionally been more used in the PBPK side for pediatric population modeling or DDI modeling or hepatic impairments. Now, coming back to the layout of this template, you can see in blue, that's the overview of the modeling strategy. There's a large number of different parameters that were already covered in detail from other speakers this morning that whoever is submitting this template should be thinking about. The virtual bioequivalence population modeling should be parameterized and should be described in detail, including inter-subject variability. Again, here, there's no big magic. Rather, this is a framework. So it talks about sensitivity analysis. It talks about calculation of model performance indicators, parameters, and we have not agreed on all the performance acceptance criteria, but I think what I'm hearing between this conference and other conferences is about 20% for PBBM models where independent studies are used. If you're running a carefully controlled bioequivalence study, then 10% is certainly acceptable. But if you're pulling in food effect data or human anatomy data or data from other studies with control arms, I think we'll probably end up closer to 20%. And this slide here caused me a lot of headache, okay? And that's, you see that in the title, okay? I had to put in the title, Kümmel Model Risk Assessment. This is a possibility, but not aligned. And the reason is because this is something which, when you give it to CMC or when you give it to some companies and you talk about the model risk assessment, if it's not been established, there's some reluctance to just use it. So what I want you to walk away from is that when this template is going to be rolled out and we have to talk about the model risk, it might be going the way that you have to describe the risk in sentences or words or paragraphs as opposed to the grid. I personally like the grid, it's my personal opinion, but there is some reluctance about, now what does this really mean? Okay, what does the 3 mean or what does the 4 mean? So for those of you who are not familiar with it, the numbers indicate the model risk. The model risk is a function of 2 parameters. One of them is what happens if the model is wrong, how severe is the consequence, and the other one is the model impact. What decision are you making? So if you waive a study and don't run the model, if you waive the study and don't run the study and only use the model, that's high impact. And if you just inform the study ahead of time, that would be considered low impact. So it would be interesting to see where we'll end up in 1 or 2 years. I personally like the grid. I think it makes sense. But I can see companies having a little bit of reluctance, particularly on the CMC side. I don't need to describe this here. And then there's a conclusion section. I wanted to come back one more time to the FDA guidance, the FDA draft guidance on oral PBPK applications, because if you're new to the field, or if you just want to know what the thinking was from 2020, but I think it's still valid. If you go to the docket and there's comments from industry, I know Satara put comments in, I know we from Merck put in, IQ put in, I don't remember all the different companies, but if you wanted to hear the thinking on the PBPK guidance for oral biopharmaceutical applications, you can find them there. And one of the things you find in there is this particular table, which talks about, you can see in brown there and in blue, the criteria for model validation that we had thought about from IQ. And it's depending on the use, and it's depending on what type of decision you want to make, but you'll find a lot that the PBBM acceptance criteria you see here are often 0.8 to 1.25. So that's because you're talking about drug product quality and you're talking about dissolution data, right? You will remember when you do DDI studies or you do some other things, you can talk about sometimes within 1 to 2-fold. But here, again, typically you end up 20% or 0.8 to 1.25. It's a very extensive list. I can't go through it here. I showed this at the workshop as well. There was a recent example from 2023 from my legacy Novartis colleagues. They had used favipiprant, and just like only look at the big picture. You have a model strategy. You lay out what you want to do. These colors, somehow they always show up in the publications these days. So there's a model setup, there's a model qualification, and then there's a model application. And you have to justify how you qualify your model before you can use it at the application stage. I just mentioned here the favipiprant is a pseudoion. There's a lot of clinical data. The specific thing here was there was microdose IV data, and I still see a lot of examples here with some type of IV data, either IV infusion or microdose. At some stage, you're going to run into projects where you just don't have it. Okay, and I think that's one of my passions, actually from day 2 from the workshop that we talked about earlier. There will be a 3 in the paper which talks about what to do, how do you generate human IV data. But here we had microdose IV data, and dissolution models are just a standard Z-factor on Weibo. And this is what I think is, we're talking about the context of use and the model credibility assessment, where we have first the question, we have the context of use, we have the decision consequence, and then we have what happens if the model is wrong. Okay, you can see here the clinical data. There was a lot of clinical data, 450 milligram, 150 milligram, multiple population study arms, because it was a phase 3 trial, so there was no shortage on the data. And basically, this is all the input parameters that are specified. I think anybody who does PBPK should use a template like this. And then you come up with some specification. As you can see here, we set the specification at the Q80 dissolved after 60 minutes. So the bottom line is, when you have this new template, the 25-page Word file will spell it out for you, and you can choose to use it, or you can choose to not use it. But I personally believe it will be helpful for industry, it will be helpful for regulators, because they'll see the same information for multiple companies, for multiple compounds. It's going to be efficient. So that's all I really wanted to talk about. You can see the timelines here. You can see there's a consolidated comment happening this month, and publication is scheduled or planned for September. So stay tuned, and if you have any comments, the slides will be posted. You can send the comments to myself or Greg, and there's several other people who actually did most of the work there on the title slide. So thank you.
Thank you for our session to the speakers to keep the time to the right.
I guess first off, do we have any questions from the audience that want to start?
Okay, guys, now is your time to ask your questions. So please think of this like your free consultation session. Any other questions from the morning session that people want to ask?
I think everyone's still full from lunch. Oh.
All right, see, that's why I'm in the audience here.
Thank you for your very nice talk, and I was actually interested in your approach. So I'm Annie DeGroot from Epivax, and we do immunoinformatics and immunogenicity modeling, which is very different from what you guys do. Still models. So I was kind of thinking about the template approach that you guys developed and wondering how you came to pull that together. Was it an academic initiative or was it an industry initiative?
Greg, for you.
It was an industry initiative with a partnership with some regulators. Yeah. So there are some regulators right now that are involved in making suggestions. There's about, led by industry, and there's about somewhere between 20 and 30 people from representing different companies. And the idea is that there's only one company representative participating, but they're kind of getting feedback from individuals within their company. There's also software suppliers involved.
I'm just wondering, I mean, here's the Center for Complex Generics. I mean, was it also facilitated by a group that kind of convened everybody? Because you kind of have to do that.
The approach that we took was to say there was 187 people that participated in the August workshop. They all had a common foundation from listening to the panel discussions as well as listening to the breakout sessions. So if they were involved with that University of Maryland workshop, then by default, they had an opportunity to participate in the process. And University of Maryland is the one that's kind of helping to hold it all together.
Actually, I'd like to add something to this for the biologics, because the concept of a safe space, a bioequivalent space or some type of safe space, right, is not just applicable to small molecule dissolution data, right? It could be applied for many different things. Maybe in biologic space, it could be % of high molecular weight species or something like that. So the reason I also share this interest is because I get asked by my management, Merck is heavily invested in vaccines and biologics, when are we going to apply this safe space also to biologic modality? So that might be something which we have to cover in a different complex generics or other workshop. But
I'm going to clarify, we work on peptides, which are a drug. They're also immunogenic.
Yes, and that I saw, I don't know if Jim is here. Jim has a workshop there later this year in October, I think.
Yeah, this fall there is a CRCG workshop on immunogenicity, and I think the group that's putting it together is just starting up, so we'll make sure that topic is at least brought up.
Hi, Michael Eckstadt with In Silico Trials. A question for Dr. Patel. You raised about 6 or 7 questions in your talks, many of them several times, and I think they're very timely and very key. And I'm wondering if you can give us a perspective, particularly on the part of the question about sufficiency. So if perhaps you can give us your view on what is sufficient to demonstrate whatever that is.
I guess that's the most difficult one you picked up. I think generally, if you are submitting a model as part of application, the sufficiency lies with the applicant. They have to demonstrate that the model is sufficient enough for the purpose. So maybe similar thing can be applied to MMF, but In other words, that means the regulators would have to have resources to evaluate those MMFs in that much detail. So again, I don't have an answer, but probably we can discuss it. So currently, when somebody submits PBPK model as part of an application, probably the burden is on the applicant to demonstrate that the model is sufficiently validated or verified for the given question. Probably the regulators would review whether they agree with that sufficiency or not. But again, I don't know if anybody has any other comments on it. But probably that's something we need to try and find out what would be sufficient and how would we describe it.
The short answer to that question is it depends. Depending on the context of use, the sufficiency may differ, right? Depending on what question. I agree with Nikunj. You have to show that it is sufficient to answer and to address the question that you are asking, and that may be different for every application.
I think maybe Dr. Kerwash, if you could provide, your perspective from regulatory MHRA on like how the context of use and the evidence and totality of evidence fit in with the framework of MMF.
So I would like firstly to comment on the template because I'm regulator and but also is it better now? So I would like to comment firstly on the template because I'm a regulator but also part of the harmonization effort through the ICH. We consider presence of template is actually something positive because the template would facilitate the rational use of modeling starting from planning, conducting, and reporting. So we support this, and it would also facilitate the communication of these models between pharmaceutical, academia, and regulators. So that's really supported. But the available template now is the only available from this PPPM applications, and it's not agreed with other disciplines, including the PPPK and POP-PK. So we would like to see more templates, and we would like to have this effort will ultimately lead to one unified template that could be used for different applications. But until now, the templates would be depending on the context of use. And back to the question about the sufficiency of the data, as mentioned, all the sufficiency in the data would be dependent on the context of use, question of interest, and how the model would be used to support a decision, a regulatory decision. So we would consider this in that the data might actually be derived from modeling, but it might also be derived from other non-modeling evidence. So we consider usually as a regulator, we consider the modeling evidence to support the regulatory decision as well as other evidence, and we call this the totality of the evidence that is used between modeling and non-modeling data. I hope this will answer.
Thank you. I think Dr. Pauly?
Yeah, sure. This is for Dr. Ahmed, who I think is online.
Yes, he is.
And so Dr. Ahmed, you're from Dr. Reddy's, and Dr. Kalipara actually presented earlier this morning. So and if I understand, Dr. Reddy's has modeling capabilities. So I'm just kind of curious, as a generic company with modeling capabilities, what do you see the biggest advantages of a modern master file?
So thanks to James, and first of all, I would like to thank CRCG and FDA for this wonderful initiative. Hope I'm audible.
Yes.
Okay. So as I said, see, from a generic industry perspective, one of the greatest thing which we see is that it will encourage use of modeling is because what we see during when we submit an NDA, ANDA application, and during its review, we get some comments or some deficiencies which we want to address using modeling. One of the major inhibitory factors is that our business is little worried that the goal date may shift if we submit near the in between the review process. So if we submit the MMF earlier or well ahead at the time of ENDA, then what would happen is that agency is already aware of the model, and if there is any deficiency or query, the review process can be faster. So from a generic industry perspective, we look forward, or what we see is the major point being the reduction in the review process, and it can also help us avoid doing some additional studies which may be asked as a part of deficiency. So this is what we are looking forward, and from a industry perspective.
Maybe just a follow-up question. So, do you think a modern master file will be more helpful for your company for scenarios where you have internal capability to do the modeling, or where you don't have the internal capability for more complex products, for example?
So I think it's primarily for both. See, first we are looking more from a oral solid perspective, where we want to kind of wave off either FET studies we talked about, or we discussed about the gender difference, or setting up dissolution specifications. So these are some things from a capability which we have in-house and which can benefit us in the near term. But since the scenario or the future is on the complex generics, where we are maybe trying to develop the capabilities, and we are not on the where we want to be. We are still on the learning curve. But yes, our journey on complex generics has just begun, I would say, and I think it will definitely help us in future. So I want to answer it in two parts. That in the short term, it will definitely help us in addressing the deficiencies, queries, or getting approvals for primarily for the standard generics oral solids. Moving forward, I think this can be extended, and we want to extend for the complex generics, where we want to develop the capabilities. So it will be a mix of both, I would say.
Okay, so thank you for that, Dr. Tausif. And there's a question over here.
I have the mic. Hi, this is Arindam. I was monitoring the chat, online chat, and we have two questions during the couple questions during the presentations, although this can be addressed by any of our panelists. So one question was during Mr. Greg Rullo's presentation: might blockchain technology be used for any model that gets accepted for the file to trace down all downstream reuse and adaptations, so the life cycle can be seen and used for further to decide about the additional adaptation? The question is, might blockchain technology be used for any model that get accepted?
Yeah, hi. Yeah, there is a recent grant that not me but the others at Satara are working on. So it's not like a blockchain, but they're trying to develop a template of end-to-end VBE, or product quality assessment. So there, say for example, if you change your in vitro dissolution data, you probably change that piece, and that might automatically update all the remaining steps. So that might simplify the modeling pathway, but I don't know if that's what they call blockchain, but something can be done. So probably everything can be connected, and if something is changed, that could probably translate into the other one, but that probably can be a useful, so it would save some time. I don't know if any other interpretation of blockchain.
It's not, sorry, it's not something I'm familiar with.
Okay, yeah. There was another question during Dr. Sumin's presentations, it could be answered, but probably partly answered also. Given the some parameters are specific to the software tool, could you please comment on how they are handled in MMF?
I believe they'll be handled case by case because the purpose of the model will be different for each application. It could be for, let's say for a PBBM, it will be expanding the acceptance criteria, or like if it's for particle size distribution specification, like widening of the specification. So in that case, I would say it would be handled case by case. For the software, I think right now the drug product part is pretty separated from the rest of the model. For example, the disposition model, whether you use the compartmental approach or the PBPK approach, that part can be standardized. And the drug product, it really depends on the MM holder, like how much of the information was shared. So yeah, it will be handled case by case.
Thank you. Thank you so much.
Other questions?
So Andy Hooker from Uppsala University in Sweden. Forgive me if this question was already asked. I was on a phone with my family before. I was just curious about the sensitivity analysis that was mentioned as this template. And I don't want to make this a discussion about PBPK models, but I'm just wondering how you guys are thinking about handling sensitivity analyses in a model, in an MMF. Almost all of the pictures that I saw that were being shown in these presentations, there was single curves, and then that those single curves should fall within some bounds. But the single curves have uncertainty, obviously, but they're not being demonstrated. So I'm wondering how you guys were thinking about that in terms of a MMF.
Thank you, Andy. I guess this could be answered by a number of different folks. I think it's very relevant.
For the Z-factor sensitive analysis I showed in my presentation, I believe you're right. I agree that we need to use a virtual B study, like simulate with the population to properly address the variability. But the problem here is we're not expecting, it's unrealistic to ask the modeler who made the model master file to like share everything. So in my case, I believe if they can, at least if they can share a bottom, like a lower bound and upper bound, like the applicant will know it's somewhere in between. And what they will see is just the upper bound, lower bound of the Z-factor instead of the, let's say the Cmax or AUC associated with that Z-factor. And for their product, for the model master file holder, for their product, they will see the Cmax and AUC, but not the Z-factor associated with it. So in that case, it will minimize the identifiability. That's the purpose for that PSA.
For the general question, is the PSA sensitivity, variability, I don't see it as a specific problem of model master file, right? It is a problem of the model development, validation, and any subsequent applicant who will want to use that model will still need to do that additional evaluation for their specific application, for their specific drug product. So I think it's not really just the model master file, it's the use of the model itself as well.
Yeah, thank you. I would also want to add some comment on that. I think for the MMF, I give an example on the methodology. So the MMF for evaluating, for example, the impact of particle size distribution on BE, there was a potential MMF to provide a successful case, and then that can be considered as a validated MMF methodology. And in that case example, sensitivity analysis was used for evaluating the impact of particle size distribution on drug exposure. So the sensitivity analysis actually we can use that to identify a safe space or upper bound or lower bound of the PSD, as mentioned by Yunming. That is a kind of methodology considered. Thank you.
Laura? Yeah, thank you. Actually, I would like to share with you an ongoing initiative within EMA. And just tell me whether or not you think that we might have a sort of bridge with this project related to modern master file. What we are busy with now is trying to use AI-based approaches to construct a sort of repository of models related to particular questions. Like one of the questions can be, you have the same active moiety, different formulation. How do we actually use a model-based approach to approach the sort of waiving a clinical study based on this and that? And then the idea is really digging into what have we already seen within the regulatory submission. So this is applicant independent. This is really an initiative coming from us. We are going to dig into what we have already seen in our system, and there is a very good, actually, voice to Patel's presentation earlier today. Go into the literature, see all the conflicting sort of, and they have conflicting sort of model and parameter values and structure, tentatively doing the same thing, which we know they want to be doing. And then we just want the assessor to be aware of the all alternatives, and at some point, I don't know where we will be landing, try at least to inform their decision also based on some sort of precedence. So I see some commonality because we are all trying to address inconsistency that might also be due to the fact that some are informed, some are not. But we take it differently because the model modified going from a sort of voluntary from the applicant to use that particular model. Whereas our initiative going from some sort of the regulator try to inform themselves on what is out there and decide. And it will, I suspect that it will happen that at some point we'll say that you wanted to use model A, but we think that there is model B, C, D that is more relevant for what you're trying to do. Just putting that out there and tell me whether you see some bridge or not. Thanks.
I would like to comment on what Flora said from also a regulatory point of view. That's really relevant because we've seen pharmaceutical companies coming for scientific advice, and we encourage them to use a specific type of modeling to support a specific decision. So if these are available beforehand and at the pharmaceutical, they are available for pharmaceutical companies, that could really shorten, that could really stimulate the utility of these models. And also it would increase the acceptability of these models between different regions, because we would use one set of models or number of models that are available for scientific companies to support their drug development program, which could actually significantly reduce the drug development program and increase the availability of medicines at a shorter periods of time.
I really like the idea that you are doing in EMA. I think probably, just hopefully you publish it or you present it to people, what you found. One interesting thing probably I think can be looked at is if you rejected a particular model from an applicant and you ask them to do a study, can you go back to those cases and see whether when they did the study, whether it complied with the model prediction. I think that itself would be a good learning, probably from regulatory point of view, to see whether, you would realize that, okay, well, you should have accepted that model while, it went to the study. So I think it might be a good, I don't know how much AI can do that, but probably, maybe some human involvement might be needed, I guess.
I think we have one question in the audience. I think this will probably be our last question.
Oh, yeah. This is Stefan Ouyang from University of Macau, the city of casino, but I don't play gambling. Yeah, so my question is everybody talk about the PBPK modeling or something like that one. However, in the MMF and another type of the modeling, such as the AI and machine learning, how it is this kind of modeling, how to, what is the rule and or what's the difference to build the, to prepare the MF for AI machine learning model? Yeah, thanks.
You want to take that? Maybe not our expertise up here with AI and ML. What? Okay. It's a good,
I just give a comment. I think in the just finished ASCPT conference, Dr. Liang Zhao gave a presentation on how we use the AI, but not specifically for MMF, but for the model-based evidence, how we can use AI to inform model-based evidence to support the drug development. Yeah, if you are interested, you can go to check that presentation.
Hi, I'm Sujata Santi from GSK. So the question I have is, we've talked about SimSip and these models very well established. What is an MMF going to do that we can't already do with the SimSip software, for example? It's very well established, right? And anything we do now, the questions you asked, will we just make it slower? There'll be version control. So I'm struggling with what is the value that MMF is producing in situations like that?
That's a great question. Does anyone want to take it?
I think the part of the MMF model that would become valid or applicable in that point is the drug and drug product specific parameters, especially the drug parameters, right? The model for the specific drug, because Gastro plus SimSip and so on give you equations, give you physiological parameters, but do not give you a validated model for any specific drug product. Well, do for some because the libraries exist, but not for the one that you have in development right now, right? So that's where the MMF would be more for the applicable
Yeah,
ones.
and I guess where I was thinking is that's going to be a drug product specific file, so you use your base platform as MMF, which I can use now without the MMF. I still have to put in my drug product parameters, and that's a specific file review process. So that's where I'm struggling. What is MMF doing here?
I think that's where I believe Tycho also tried to bring that up was harmonization. Are we going to get a agreement between different regulatory agencies so that once you have that model file and got reviewed by agency, would the next one accept it?
Yeah.
I have a follow-up. My name is Joga Guburu, University of Maryland. I'm trying to wrap my head around MMF myself, and what I see is a mixture of two completely different topics together. Please confirm if I'm mistaken, I would be happy to accept. Science of model development and MMF, the way I see it, I have experience with DMF, there's nothing in common. Let's leave the science part out. The MMF only is helpful, according to my understanding, drawing analogy from DMF, that once you agree, whoever that is you, on the model, then can we mix it in the municipal water supply that others can use? That's the notion of MMF. So you don't ask questions whether that model is validated, where is the uncertainty, all that stuff, it's done. This is a logistic maneuver of regulatory submission of your model such that, again, I don't know if that's where the time is wasted of re-reviewing the model every time. Is that the problem we're solving? I don't know what problem we're solving. So to me, if we can focus on the MMF as a submission that can be reused without having to be reviewed again, would be much more productive, in my opinion. Otherwise, you would have several of the audiences going back with this uncertainty of what the heck are we talking about here?
I have one comment. I think referring to the discussion in the morning panel session, I think Floria mentioned that they would still want to look at the model again for the new context because the same applicant is not going to submit the same model for the same question. So if the review of the model or MMF is going to be again for a new context, then going back to the question, it's similar to what we are doing right now. So I don't know what.
I think they're great questions that we're going to delve into this afternoon, hopefully. But I think that's time, right? I think I want to thank everyone, our panels, for a great discussion.
Thank you, everyone.
And welcome to the next session.